Okay, so this talk is based on the work that we are doing in my lab, and my PhD student Deepan is doing a lot of this work and coordinating it along with a bunch of other students who are interns and doing master's projects and things like that. So the theme of this talk is computing with rhythms or computing with oscillations. That we say that there is a need for a third category of neuron models. Actually, it's not a neuron model, but a neural ensemble model, but a model of neural code, right? And so there we need to describe it in terms of oscillations. So we say that if you construct networks using such models, they'll be very powerful. They'll be able to explain a lot of effects, which could not be easily explained by the two previous approaches, which is the rate code and the spike neuron code. So okay, so this is a general talk, so I'll quickly skim over some of these basics. Right, we know brain is a network of neurons, and they communicate by exchanging these electrical signals, and these are the spikes that if you inject current into a neuron, it produces these action potentials. So the big question is, what is the neural code? And here again, like I said, there are two schools of thought. The spike code people say that it's all in the spike times. Okay, when do the spike occur? When does the spike occur? Right, and the other this thing is, if you look at a waveform like this is a voltage waveform. The information present in this waveform is simply in the form of the spike frequency, right? So if you extract that from this waveform, you will get a smooth signal which goes from a high value to a low value. Okay, so this is the rate code and this is the spike code. So these are the two schools of thought. Thing is, both of these approaches refer to a single neuron. Okay, so output of single neuron produces spikes, and that can also be looked at in terms of the rate. Typically, what happens is in practice, right? The spike neuron models are considered more biologically realistic. If you are talking about a serious model of a brain, right? Of model of the brain or brain function, use networks that have spike neuron models. So they are called spike neuron networks. Whereas rate-coded neural networks, like for example the McCullough and Pitts neuron model, people also use other kinds of nonlinearities. For example, in deep learning these days, typically people use things like ReLU or rectifying a linear unit, rectified linear unit. So, but anyway, rate-coded neural networks are more commonly used in the current deep neural networks and AI kind of a domain. See, the rate-coded neuron models, again, you have the sigmoidal neuron, which is what we discussed. The McCullough and Pitts neuron is a sigmoidal neuron. And I was just calling it the binary neuron, but basically it's the same kind of thing. Then you also have the ReLU neuron, where the nonlinearity is not like a sigmoid, but is given like this. It is so for y equal to x, for x greater than 0, and y equal to 0 otherwise. Okay, so that is called a rectified linear unit or ReLU. And the other, more complicated neuron models which have memory property. One good example is the long short-term memory block. Right, and deep learning and AI are very powerful and quite successful. So therefore, they can even reproduce a lot of effects, a lot of intelligent behavioral patterns that are exhibited by humans, like complicated challenges in image processing, computer vision, and speech recognition. They have been solved quite effectively by deep learning and AI type models. So people tend to think that brain works like this, or at least of late there is also some very successful studies which show how deep learning neural networks can be used to explain a lot of brain functions. So that makes the the situation, the literature quite interesting. Yeah, so if you look at the history of neural networks, so also there's a little political angle to this. Neural networks began maybe in the 1940s with the McCullough and Pitts neuron model. Then in the 60s, this guy called Frank Rosenblatt came up with this learning neural network called a perceptron, which was able to do some very interesting functions. And we'll talk about perceptron in the next class, or next couple of classes. And that was extended further in the 80s to multi-layer perceptrons. And then they were applied to a lot of problems in psychology, in pattern recognition, and so on. So they were become very successful. But the effort slowed down in the 90s because there was too much hype, and networks didn't seem to be that powerful. So the hype kind of fizzled out in the 90s. And in the 2000s, again, in 2006, there was a paper by Hinton and others on deep learning. So that revived the whole field, and they became quite popular. But the political angle here that I was talking about is originally AI and neural networks were in rival camps because the AI community thought that you can explain intelligence in terms of a bunch of rules, right? If the rules are complicated and comprehensive, right, the behavior that they generate will look complex and look intelligent, but it's all a bunch of rules. So that was the AI stand. Whereas new network community said that it's not rules, it learns, right, by experience, and it kind of embodies it, all that data in the form of a network structure. And there is no explicit rule here, it's all there in the connections. So that led to the revolution, the connection is revolution. So they belong to rival camps. And there was a tug of war between the two communities and two approaches for many decades. Until the 2006 paper by Hinton, when it became like neural networks were a clear winner. That means they won this war with AI. Then AI people said, no, it's all part of AI. It's all part of this umbrella thing called AI, and that's how it looks like. This is not actually AI, but this is neural networks. Anyway, so a lot of developments took place, and the deep learning revolution took place after that. And so basically, in deep network, you look at very large networks with many hidden layers, compared to traditional neural networks, which typically deal with only one hidden layer. But there's nothing fundamentally new about deep networks. We'll discuss this also later on in the course. So if you look at spectrum neuron models, we talked about leak integrated fire neuron model, one variable, in Zizkiewicz, two variables, Austen-Huxley, four variables, and biophysical neuron models can have very large number of variables. I mean, you can have like a few hundred differential equations to describe a single neuron. And that's quite possible. There are softwares which will let you do that. The question is, why do you need it? I mean, that's a different matter. Okay, so we have seen all this, Austen-Huxley, we have seen all this, biophysical, we have seen this. So now we have two extremes. Right, on one side you have the rate-coded neuron model, right, with sigmoid neuron and LSTM and that kind of thing, and people complain it's too artificial. On the other side, you have detailed spike neuron models. There again, you can go way beyond simple spike neuron models, and look at biophysical models with dendritic arbors and etc. etc. Okay, now the question is, what is the right level of abstraction? Right, both sides have their own advantages, because if you want to study, for example, how does a neuron's firing pattern changes if you introduce a mutation in one of the ion channels that are expressed in that neuron. That to answer a question like that, you need to go all the way from molecule to whole cell, right? You need to understand how, if there is a change in an ion channel, how does the dynamics of the ion channel change? Then you have to write the full voltage equation. C dV by dt is equal to, write all the channel currents. You have to write, and so then if there is a mutation in particular ion channel, then you have to find out how the channel current changes as a function of voltage, right? And then rewrite the equation. So all this is very difficult. You have to go all the way from single molecule to the whole cell. But if you want something like how do we recognize images, how do we recognize sounds, right? Then you don't need to worry about the dendritic trees and all that. You can manage with much simpler neuron models. Okay, the question is, what is the correct level of description? What is the correct middle term? Now here, since we are engineers and this is a technology institute, I would like to give an analogy to answer this question. If you take the problem of fluid mechanics, right, an important problem from fluid mechanics, for example, you want to understand study an aircraft wing, right? There's wind blowing on the wing, and you want to understand the kind of forces that and pressures around the aircraft wing. So how do you do that? You don't try to solve molecular dynamics simulations and find out the actual impacts of single molecules on the wing and things like that. I mean, probably you have the computing power to do that, but it is too brute force, too crude, right, as a scientific method. And also it might take a very long time to model all that, for a large object like a aircraft wing. So obviously engineers don't do all that. we use, we just look at the wind as a fluid, right, and use write down the Navier-Stokes equation and calculate the pressures and stresses and sharing forces and all that stuff. So, but then what is this thing called fluid? Fluid is not molecules. So, fluid has molecules in it, but when we mathematically talk about fluid, we talk about a fluid element, right, of infinitesimal volume, like dx dy dt. And inside that volume, we know that there are a bunch of molecules, but we never refer to the molecules. We refer to this kind of a collective thing called fluid, and talk about its pressure, its density, and temperature, and viscosity, and all that. But we never mention molecules. What we are dealing with is a macroscopic thing, an average quantity, a statistical quantity. Right? We never go to single molecules. And but only thing is, just because you're talking about an average thing or a statistical thing, your description is not vague or fuzzy. It's very precise, very accurate. Navier-Stokes equation is not fuzzy or vague. It's very accurate. It's very accurate. But it's an abstract thing. It's not dealing with single molecules, and that is why it is become. It is so powerful. So if you take this analogy even to the neuronal case, the brain case, the problem of describing brain function. One big problem is what is the right level of abstraction at which you want to describe brain function? Should we go all the way to the single molecules or single dendrites, or is it single neuron, or is it something else? This is a big question, and there's no simple answer to this. So there's one school of thought that believes that the traditional school is that the single neuron is the basic functional unit. That's a fundamental building block of brain's computation. In fact, that idea came, it's almost a century old. I mean, if you look at Raymond E. Cajal, right, who is called the father of the neuronal doctrine. He said that the neuron is a functional unit of the brain because anatomically, definitely neuron is a functional unit, basic unit, right? But functionally, if you want to describe brain function, if you want to write down the mathematical equations, what is the most fundamental unit? Is it neuron or is it something else? So there is another school of thought which says that that function unit is not a single neuron, but a bunch of neurons, what is called a neuronal ensemble, a population of neurons. Now, therefore, the signal of such an ensemble is not a spiking, a train of spikes, but a more average signal, which looks like what you see in this picture. So, if you are measuring the activity of a neuron, at, like by poking an electrode, right? Sorry. If you poke a neuron with this microelectrode like this and measure it, you see the spikes. But if you don't go all the way to the single cell and just kind of poke the brain tissue with a slightly blunt electrode, thicker electrode, then what you measure here, because it's an ECS, it's not penetrating any neuron. What you measure here will be some kind of an average activity of all these fellows. So average activity of all these fellows. And that signal won't look like spikes. It look like a smaller signal like this. Okay, and that is called the local field potential, LFP. Okay, so here again, sorry, so this is not, so this is the actual signal, but if you low pass filter it, you get a signal which looks something like this, smoother signal. That is local field potential. And if you high pass filter it, right, you can pull out these sharp, sharp spikes, and these correspond to your action potential. Okay, so there is a school of thought which says this is a very interesting signal. Let us model this. Let us study this. Let us describe brain function in terms of these components and not this. But this is one school of thought, and I strongly believe in this approach. I feel if we explore this approach more extensively, by developing the right kind of mathematical tools, right, it can really push the frontiers in neuroscience and in theoretical neuroscience. Okay, now let us talk about these brain oscillations. So, for example, you might have heard that if you record brain's activity by putting electrodes on the surface of the head, right, on the scalp, right, you can pick up this kind of electrical activity of the signal, which is called the EEG or electroencephalogram. Okay, so this was recorded almost more than 100 years ago by a guy called Hans Berger for the first time. And here, if you analyze the signal, look at its spectrum, and people find that it's not like a flat spectrum. There are certain peaks in it. and people have given names to these peaks. But again, these are not like cast in stone. It's very hard to draw clear boundaries. But anyway, we have to give some names and draw some boundaries for explanatory power. So if you're starting with the lowest frequency, some of the lowest frequency components, from 0.2 to 3 hertz, these are called delta waves. And this is the kind of activity you see in dominantly in deep and dreamless sleep. Then you have theta waves, 3 to 8 hertz. These are important for strengthening synapses. And theta waves are also seen in navigating animals. So if you look at rats which can run mazes and escape from mazes and things like that, during that process you will see a strong theta wave emerging out of its hippocampus. And hippocampus is an important organ in the brain involved in spatial navigation. So similarly, alpha wave is another important, very ubiquitous kind of a wave found all everywhere in the brain. This is from the 8 to 12 hertz. And this is found when you are in a calm, relaxed state. For example, the kind of state that you are in right now, when you are hearing this lecture. Then you have beta waves between 12 to 30 hertz. Then beyond 30, it's gamma. And gamma is like everything above that. It's a very vague, kind of a very wide sweeping kind of a band. So here people talk about low gamma, high gamma, and all that, but basically people are clueless, and so this whole thing is called gamma. So these are the main waves, and so if you want to model brain's activity, not in terms of spikes, but in terms of the smoother signals, right? The advantage is you can use all the signal processing techniques from engineering, from electrical engineering, right? You can use ideas of transfer function, spectrum, and whole powerful bag of tools from linear systems theory, right? And so that completely changes the scenario of how you describe brain function. So this is what we are trying to do along with a bunch of collaborators. That I feel that there is a huge potential to even revolutionize computational neuroscience. If you look at brain function in terms of these LFTs and these smoother signals, and borrow ideas from electrical engineering and systems theory and so on. Okay, so here I'll just kind of pause with the quotation from Bazaki. So Bazaki is a pioneer in these kinds of studies. He's an experimentalist who studies the brain rhythms. He describes brain function in terms of brain oscillations. So he says that synchronous activity of oscillating networks is now viewed as the critical middle ground, right, linking single neuron activity and to behavior. So you want to go all the way from single neurons, even to subcellular components, all the way to behavior, what you see outside. If you want to do that, these oscillating units, these networks, seem to be a good middle ground. This is what these people say in a science paper in 2004. And for that, you need a different kind of mathematical tools. And we are trying to develop one such tool using the Hopf oscillator. I don't know if you remember, we talked about the Hopf oscillator as one of the possible neuronal models. So we'll start with that and present some interesting network models right in the next class. Thank you.